Good day, I'm Rocher Cruz, and today I'm going to present to you my Science Quarter 3 Project 1. What is DNA? DNA, or deoxyribonucleic acid, is a long molecule that contains our unique genetic code. Like a recipe book, it holds the instructions for making all the proteins in our bodies. DNA, along with the instructions it contains, is passed from adult organisms to their offspring during reproduction. Structure and parts of the DNA and their chemical composition. A molecule of DNA consists of two strands that form a double helix structure. DNA is a macromolecule consisting of two strands that twist around a common axis in a shape called a double helix. The double helix looks like a twisted ladder. The rungs of the ladder are composed of pairs of nitrogenous bases, base pairs, and the sides of the ladder are made up of alternating sugar molecules and phosphate groups. Molecules of DNA range in length from hundreds of thousands to millions of base pairs. The smallest chromosome in the human genome, chromosome 21, has around 48 million base pairs. Each DNA strand is composed of nucleotides, units made up of sugar, deoxyribose, a phosphate group, and a nitrogenous base. Each strand of DNA is polynucleotide composed of units called nucleotides. A nucleotide has three components: a sugar molecule, a phosphate group, and a nitrogenous base. The sugar in DNA's nucleotides is called deoxyribose. DNA is an abbreviation for deoxyribonucleic acid. RNA molecules use a different sugar called ribose. Covalent bonds join the sugar of one nucleotide to the phosphate group of the next nucleotide, forming the DNA strand's sugar-phosphate backbone. A nitrogenous base is an organic molecule that contains nitrogen and has the chemical properties of a base. There are four nitrogenous bases that occur in DNA molecules: cytosine, guanine, adenine, and thymine, abbreviated as G, C, G, A, and T. RNA molecules contain cytosine, guanine, and adenine, but they have a different nitrogenous base, uracil, instead of thymine. The sequences of nitrogenous bases on two strands of a DNA molecule are complementary. The sequence of nitrogenous bases on one strand of DNA molecule's double helix matches up in a particular way with the sequence on the other strand. Adenine pairs with thymine, and cytosine pairs with guanine. Why do nitrogenous bases pair in this specific way? The bases on each strand are joined to the bases on the other strand with hydrogen bonds, but different bases have different chemical structures. Cytosine and thymine and uracil in RNA are pyrimidines, containing one ring. Adenine and guanine are purines, containing two rings. The pyrimidines pair with the purines. Cytosine and guanine form three hydrogen bonds, and adenine and thymine form two. Specific sequences of nitrogenous bases that code for particular proteins or regulatory RNA molecules are called genes. Each strand of DNA is like a recipe book for synthesizing proteins. Certain sequences of nitrogenous bases along the strand encode particular RNA molecules. These sequences are called genes. mRNA molecules transcribed from genes are translated into proteins later. Chromosomes can vary widely in their number of base pairs and genes. The longest chromosome in human cells, chromosome 1, is around 248 million base pairs long and has between 2,000 and 2,100 distinct genes. Chromosome 21, the shortest human chromosome, consists of 48 million base pairs and contains between 200 and 300 genes. Overall, prokaryotic cells have shorter chromosomes with fewer genes. For example, the bacterium Carcinella rudei has only 159,662 base pairs and 182 genes in its entire genome. Although genes get most of the credit for what DNA does, they make up only about 1% of DNA in humans. Genes are separated from one another by sequences of nitrogenous bases that don't provide instructions for RNA synthesis. These are called intergenic regions. Even within genes, there are regions of non-coding DNA called introns. Non-coding regions of DNA are important because they provide binding sites for proteins that help activate or deactivate the process of transcription. They can also provide protection for the coding regions. For instance, telomeres consist of repetitive sequences that protect the genetic information on each DNA molecule from being damaged during cell division. Roles or functions of the DNA in living organisms. DNA contains the instructions needed for an organism to develop, survive, and reproduce. To carry out these functions, DNA sequences must be converted into messages that can be used to produce proteins, which are complex molecules that do most of the work in our bodies. DNA function. Replication process. Transferring the genetic information from one cell to its daughters from one generation to the next, and equal distribution of DNA during the cell division. Mutations. The changes which occur in the DNA sequences. Transcription, cellular metabolism, DNA fingerprinting, and gene therapy. Now that the 3D DNA model is finished, I will explain this model to you. This is the sugar phosphate backbone. This red color represents the thymine. This one is for adenine, the blue color. The green color represents the cytosine, and the violet color represents the guanine. This yellow part is for hydrogen bonds.